State University. I'm talking to you today about a pipeline I'm developing to trace the origins of orphan genes. It's very close to what Amir presented on earlier. So a common model for protein evolution is that all modern genes are derived from very ancient precursors. But with the sequencing of thousands of genomes now, we see that a lot of genes are actually don't follow that model and that the gene birth process is ongoing. So our practical definition of an orphan gene is a gene whose predicted protein has no significant similarity to any known protein in any other species. Basically, you can't really find them in BLAST. Orphan genes are typically short, simple, and usually monoexonic. And they comprise between 1 and 30% of the genes in an organism. And what do they do? So there are very few orphan genes that have actually been well characterized. My probably favorite one is a antifreeze protein in an Antarctic ice fish. There's also a gene in Rhabdopsis thaliana, which integrates signals from the environment to control the starch-protein ratio. There's also a lot of circumstantial evidence that they are involved in stress or interspecies relationships. So where could they possibly come from? Amir basically covered this pretty well for me, but the two general possibilities are de novo emergence from non-coding DNA, or perhaps they could actually be descended from ancient protein coding genes, but they've evolved beyond recognition. So the objective for my pipeline is to trace these orphans to their genic or non-genic precursors. So my approach, first of all, I choose a species of interest, Rhabdopsis thaliana. Then I find the orphans using the traditional approach with BLAST. I blast all genes in Rhabdopsis thaliana against other things, and find 902 orphan genes that have no significant similarity. Then I select a set of related species within 4 members from the Brassicaceae family, and I align the query genome, Rhabdopsis thaliana, to each of these members, to Rhabdopsis lyrata, Capsella rubella, Brassica rapa, and Eutrema salsogenium. Now that I have these maps, I can map the query genes in the Rhabdopsis thaliana to specific search intervals in the target species. If you want to know the algorithmic details, see my poster. There's things missing from my, something's not showing up on my PDF. There should be, all right. There's a little gene here. If you want to see it, come see my poster. Strange. Okay, so once I have these specific search intervals, I can search within them for homologs. So first I look for a amino acid similarity of my amino acid orphan gene, my protein gene, against open reading frames in the target. If I don't find any hits there, I search for the DNA sequence against the search interval DNA sequences. If I find nothing there, then I label it as unknown. A little bit more detail. You know, if you want more detail, come see my poster this evening. But it's not showing very well. But essentially, there are several different categories of homologs that we distinguish between, which is useful in downstream studies. Also, if we find no match, we don't just give up and throw up our hands. We can actually predict reasons for that. Again, it's going to post for details. Okay, so next big thing, we take all those labels for query target pairs, and then we map them to our tree. Say for a query gene thaliana there, look at A. lyrata. We find a orphic hit, C. rubella, you map to an unknown region. Brassica rapa and Eutrema salsogenium, you map to non-genic and orphic. And then you get this ancestral vector. And look at the ancestral vectors, you can classify your genes into by origin class. We find 104 candidate genes that are quite possibly, or that could be members of a very rapidly evolving families that are conserved across Brassicaceae but aren't identifiable by BLAST. We find 167 that are possibly present in a genus but not elsewhere. We find 175 that are very likely very recent non-genic origin. And 456 that we can't quite trace yet. And we also run non-orphan pseudopipeline as a control. In conclusion, so I've developed a framework for reducing search space using a Centiny map. This can actually also be adapted for general ortholog prediction. And it is the starting place for a whole bunch of really cool analyses, which I'm planning in the near future. If any of you are looking for a postdoc position, we have, you can help me. Okay, so acknowledgements.